apparently the reason why the whole screen didn't show up last time was because it zooms in when it's video. Alright, so we're going to go ahead and review the entirety of cell biology today in one startlingly fast hour. So, we're going to fly through everything because there's a lot of stuff to cover. I also put cell signaling at the very end of this, so that way we have a chance to review it, since we're not going to have enough weeks before the test to go over anything we've covered since the DNA and genetics test. So, basic most thing about cells is that you have to have a proper surface area to volume ratio, meaning you need to have enough cell membrane around the cell to be able to move in nutrients and move out waste, as well as to perform any metabolic processes that have to happen along a membrane. So you need a large enough surface area, so cells do what they can to either maximize their surface area or reduce their volume. Ideally, if you're a smaller cell, that's fine, because when you're a smaller cell, you have a lower surface area to volume ratio. However, small cells have their limitations. You'll notice most eukaryotic cells are actually much bigger than prokaryotic cells for a reason. You can pack more functions into the same cell. And then not only do you need more surface area of membrane in order to adequately move nutrients in and waste products out for every, it's not even cubic millimeters, every little tiny volume of space, but for every expansion you have in volume, you need more materials and more energy to keep that cell alive. So if you tried to have a cell the size of this classroom, it would die very quickly. One, because things diffuse once they get into the cell. You can't have anything too far from the cell membrane, so that way nothing can diffuse to it. And you also need to have enough energy to support all of that cytoplasm. All right. So there are multiple adaptations that cells have in order to reduce to increase the surface area to volume ratio. It's either to reduce volume or increase surface area. So a very common adaptation is something like this root hair that you see coming off of a cell. So these are cells in the root of a plant, and they have these projections coming out. So you'll notice they don't add much volume to the cell, but they do add more surface area. And so that way roots can absorb more nutrients and be more effective at their function for the whole plant. You see the same thing in the small intestine of a human. So in our small intestines, if you were able to look at the tissue, if you've ever done a rat or a mouse dissection, oh, I'm sorry, you didn't get to. Last year, I know we did mouse dissections, and I had them open up the small intestine or the stomach, and they felt the inside, through gloves, of course, and it feels kind of rough. It feels like it's got texture, little nubbly things on it. Those are actually these structures called villi. So villi are these projections that stick up out of the small intestine to help maximize absorption area. And then each cell in a villus has a bunch of microvilli. So it's these finger-like projections, each of which has thousands of finger-like projections sticking out from the individual cells. And that adds surface area without increasing the volume. So cells have a lot of adaptations like that. This is a cell in the alveolus, which is the part of your lung where gas exchange happens. So you'll notice, you see that that speck right there, that is the nucleus of a cell. The cell looks really large, but it's actually got a hollow space inside. So it's got a very large surface area because it's like a balloon stuck inside a slightly larger balloon. And so it's got the inner surface area and it's got the outer surface area. So it's got a lot of space over which it can exchange carbon dioxide and oxygen for us without having to support a whole lot of cytoplasm and other molecules and processes inside of the cell. Okay? Membranes are really useful, but we do have organisms that don't have have a lot of membranes inside of them. Archaea are the kind of most primitive form of life. They are some of the simpler bacteria, and they don't really have a lot of internal organization. They have a region where their DNA tends to sit. It's called the nucleoid. They have some ribosomes and a whole bunch of other stuff floating around in the cytoplasm, and that's about it. All packaged in a cell membrane and then a cell wall. Some bacteria, which are a little more recent as far as evolutionary development, though I mean by that maybe 500 million to a billion years younger. So still quite a ways away from the first evolution of life. So you see on the right-hand side, there is a structure that looks kind of like a chloroplast. That is a cyanobacteria. It is a bacterium that is capable of performing photosynthesis. And what you'll see is that it has not detached like organelles made of membrane, but it does have these infoldings of the plasma membrane, and that is adaptation that some more advanced bacteria developed. So as you start looking in slightly more complex bacteria, you'll start to see something that looks like it's almost a transition point between prokaryotic cells with no internal membranes and eukaryotic cells with lots of organization and all these organelles that you guys had to memorize inside of them. So these are still interconnected with the cell membrane. They are not completely separate, and that's why they don't count as separate membrane-bound organelles. But as we get to eukaryotic cells, you'll notice we have a lot more structures inside. So this is just showing some of the structures that you find. This is mostly the endomembrane system, a system of interconnected membrane-bound organelles. So the nucleus, which has a double membrane layer around it, is physically connected to the endoplasmic reticulum, which is going to be connected by popping off little bubbles of membrane that will join up with this guy, which is the Golgi, and the Golgi will then release substances either through these vesicles to other parts of the Golgi or very commonly to the outside of the cell. So what these internal membranes do is they allow you to compartmentalize the cell. You can have different pHs in every single one of those compartments. You can concentrate chemicals. In fact, that's what you do in the Golgi. You build up concentrations of certain proteins, and then you're able to pack them into a vesicle and spit them out of the cell. Or you can also have increased area for any chemical reactions you want to have happen on a membrane. So we most recently covered photosynthesis and cell respiration, and for both of those processes, a lot of the molecules that are actually doing the work, producing energy or harvesting energy, are sitting in a membrane. So the more of that membrane you have, the more surface area you have actually performing your task. And we're going to see that theme over and over again. So you have a couple of examples. In chloroplasts and mitochondria, you can make proton gradients. That's a gradient, concentration gradient of hydrogen ions that you can then use to make ATP. You have the nuclear envelope providing extra protection for your DNA. Your DNA is constantly under threat of hijacking by bacteria or viruses, and so an extra physical barrier between your DNA, which you need to keep unmutated so you can stay safe and uncancered, it will help be an extra layer of protection to prevent mutations or to also prevent viruses from coming in and taking over the cell. Endoplasmic reticulum and Golgi let you not only concentrate proteins, you can stick enzymes in those compartments, and you can use them to make specific modifications to proteins, sort them, and then direct them. And you have these little vesicles, transport vesicles. They're little bubbles of membrane, and they're like little bags that are used to just cart molecules around. In fact, we saw some in the nervous system. You have actually little vesicles, which are just little bubbles of membrane, chock full of neurotransmitters, and those get released whenever a nerve cell is communicating with another nerve cell. So, there's a lot of important properties of the cell membrane that make it very important because it separates the external environment from the internal environment. That is what allows us and all living things to make sure that we, say, concentrate glucose inside of us or expel waste. We can create different environments from our surroundings. And there are a lot of complex things embedded in this membrane. We'll talk about them. Let's go on to the most important guy, which is the phospholipid. The most important things to remember: the head is hydrophilic, it is charged. It will interact with other charged molecules or polar molecules because likes will hang out with each other. If you're charged and I'm charged, we'll hang out together. If you've got a water molecule somewhere around this guy, it's going to hang out near the hydrophilic portion. In fact, that's what philic means, loving. So it's the water-loving part. On the other hand, you have hydrophobic fatty acid tails sticking off the end. And the reason why those are important is because if you stick a bunch of these phospholipids in a liquid and you shake it up, they're going to end up arranging themselves so their fatty acid tails are facing each other. That's going to make them form this kind of barrier, and if you shake up that container full of phospholipids, some of them might even form spheres. And that's going to form a bilayer. And that bilayer is important because, due to its properties, it only lets certain materials in and out of the cell. And we'll talk about that in a few minutes. You do want your membranes to be fluid. Membrane proteins and membrane lipids move around a lot, and you want to encourage that fluidity. If they stay kind of stuck in one spot, that's not good for the functioning of your cell. So if you have more unsaturated fats in your phospholipids, that's a good thing because they kind of stick out, they don't pack together closely, and they can move around more. If you have a bunch of saturated hydrocarbon tails, they're just going to stay in place. And much like with our bodies, our cell membrane should not stay motionless. Cholesterol is also good because it can help modify the fluidity. It will help to stabilize the membrane. So if your temperature goes high, it'll help stiffen it so it doesn't move so much that your cell starts falling apart. At a low temperature, it's going to keep your membrane from freezing, which is good. When you're trying to maintain homeostasis, if I stuck you in the middle of Antarctica, you don't want your cell membranes to freeze. You want them to be able to be fluid. All right. There are a lot of proteins that are stuck in a membrane, and the proteins that are stuck in a membrane determine its characteristics. You've got your base model, just a phospholipid bilayer with some cholesterol stuck in. But it's very limited in what it will allow to cross. And so if you want to, say, let water in and out freely, you're going to need some proteins. If you want to allow glucose to get in and out freely, you're going to need some proteins because these things don't cross a phospholipid bilayer on their own. So, depending on the cell, depending on the organelle, you're going to have specific proteins stuck in the membrane. There are a couple of different types, some that are temporarily bound. They'll usually bind to the inner surface, sometimes the outer surface. An example are G proteins, like with our G protein coupled receptors. Then you have integral membranes. They are permanently bound, and a lot of times they're like this guy. They stick completely through the membrane, and a very common function for them is to move stuff across the membrane. There's also the possibility that it could be like a G protein coupled receptor, and it's taking a message from the outside and transferring it to the inside. The way that these proteins are made, they're actually produced by ribosomes stuck to the endoplasmic reticulum, and from their very formation, they are stuck inside of a membrane. And their physical properties are responsible for that. So this section right here that's in red is actually hydrophobic. It will not interact with water. This part right here, though, is polar. It is hydrophilic, and it will interact with water. If you tried to take this protein and push it out of the membrane, since water will only be attracted to other charged particles and it will actively exclude nonpolar things, that protein would be pushed back up into the membrane. So the properties of the protein let it stay in place. The fact that it's got hydrophobic and hydrophilic parts. And that's a common thing you'll see in anything that's stuck in the membrane. Anything that faces the outside or the inside of the cell will be polar and charged. Anything in the center will be nonpolar, uncharged, and hydrophobic. You do have very special classes of molecules. This is one of the more important transport proteins you have in your cells. You do need to be able to regulate water intake and outflow from your cells, but water does not very easily cross the cell membrane. It's polar, so it's got a bit of a charge, and it will not interact easily with those hydrophobic insides of the cell membrane. So your cells have special molecules called aquaporins. Aquaporins are just like doorways sitting in the cell membrane that are constantly open, and they'll allow water to flow in or out. So they don't control the direction of water flow, and they don't regulate the water flow. It's always open. On the other hand, you can have other things like this ion channel, which can be gated, meaning they can be closed or opened depending on specific conditions. All right, these are some of the other classes of proteins you'll find in the membrane. So you have some things that are like carrier proteins. They're designed specifically to carry a certain molecule across the membrane. Most of these proteins that are stuck in the membrane are very specific to what they'll interact with. So this protein, for example, if it's a glucose transporter, will move glucose into or out of the cell. It will not move sucrose or fructose or an amino acid or an ion. It is very specific to glucose only. You also have some proteins whose job is to just sit there and declare, I am part of the cell. So when we talk about the immune system, you have the ability to recognize self versus non-self. This protein would be declaring, I am self, I belong in this body, please don't attack me. When you have things go wrong with these recognition proteins, that's when you have things like autoimmune diseases where your body starts attacking its own cells. Receptor proteins are really important for signal transduction or cell signaling, which was our last topic of all. And they are going to bind to a very specific molecule called their ligand or ligand, and they will set off some response. Usually there'll be some physical change in them, and it's going to cause either them to pass a signal along, or it could be that they're an ion channel and they open up when that thing binds. It depends on the specific molecule. You also have enzymes that can sit around in the cell membrane. They can be activated by various signals, and they will perform a specific chemical reaction. Just depends on which particular enzyme it is. You also have junction proteins, which will allow cells to adhere to each other. This junction protein looks like it has a channel inside of it, so it would allow direct contact between the cytoplasm of two cells. That would be like having a conjoined twin. Your bodies are sharing molecules and everything. You're not just talking to each other. There's all sorts of communication going on physically. But you also have some other proteins that can stick inside the membrane that can help you stick to the extracellular matrix. That's the outer parts of our cells. There's stuff that's going on out there, but you want to stick to a certain area. It can help you stick to other cells in the region, so that way you are a collection of cells instead of a bunch of cells floating on the ground. There are carbohydrates that are also important in membrane function. These tend to be found on the outside. A lot of them have to do with cell signaling or recognition, and you can also have glycolipids, which is where you take a lipid and attach some carbohydrates to it. I don't think I have a picture of it up here. So, like I said, these proteins are important for cell recognition and also for cell adhesion. When you have cells differentiating and developing in utero, when an organism is developing, some of these signaling molecules on the surface of the cell will help other cells determine an identity and move to the right location inside of the body. There's a lot of migrating happening during development. All right, let's talk about transport, permeability. So the plasma membrane is only semi-permeable. Like I said, your base model membrane with just some phospholipids and some cholesterols in it is very picky about what it will allow to enter. If you are small or uncharged, you can cross freely. So carbon dioxide, oxygen, small uncharged molecules. Glycerol is a fairly small uncharged molecule, happens to be one of the building blocks of fatty acids, and some of the smaller alcohols can also cross. If you are large or in charge, as I like to say, so if you're large like a starch or a protein, even sugars like glucose are too big to fit through the plasma membrane. Or if you have a charge, you can be polar like water, or you can be a full-on ion like sodium and calcium. You shall not pass. Those have to be assisted by carrier proteins in order to get across the membrane. And like I said, water, it has a bit of permeability to water, the membrane, but it's nowhere near enough to allow you to have that osmotic balance we'll talk about later. So that's why you have aquaporins. So if we're looking at molecules and them crossing the membrane, notice charged molecules and ions can't cross. Water can't cross easily unless it's going through one of these aquaporins. Non-charged molecules can get across though. Larger molecules cannot. And that's the story of what moves. Let's talk about cell walls. So cell walls are an extra layer of protection. They also provide some structural support for different types of cells, most commonly plants, fungi, and bacteria. They will also be a permeability block, so they can also have their own selective permeabilities, things they will allow inside and will not. So, plant cell walls are the most familiar type to most of us. They are made of a molecule called cellulose. They are fairly rigid. They allow plants, since they limit the volume of the plant, they allow them to take in as much water as they want without bursting, which will become important when we talk about hypo, hyper, and isotonic solutions. And they're also a physical barrier to infection. Plants don't have their own like roving immune system cells, so it's each cell for itself trying to defend itself. And also it can help just shape the plant as the plant is growing. It is secreted or released outside of the cell membrane. There are some other cell walls. So here we have gram-negative bacteria, gram-positive bacteria, another type of mycobacterium. One of the common things you'll see is this molecule, which is called peptidoglycan, and that's a very common element found in bacterial cell walls. Though I will say archaebacteria don't have it. You don't need to know anything about what archaebacteria have instead. Notice that at the bottom of each of these diagrams, you do have cell membrane. So this would be the extracellular or outside the cell space. And notice you just have a bunch of these molecules forming an extra barrier outside. You may have some proteins and such that help form a barrier to allow molecules to enter or leave. Then you also have fungi. Fungi are obviously not bacteria or plants. They have cell walls, and their cell walls are made of a different substance. It's called chitin, not chitin. And chitin is also found in some other organisms like insects. It's what makes their exoskeletons all crunchy. So when you step on one and it goes, that's the chitin. All right, passive transport, you don't need energy for it, so this can happen on its own. It is always movement of molecules from a high concentration to a low concentration. It can be important in not only importing nutrients or food and exporting waste, but as we saw with photosynthesis and cell respiration, it is also important for building like a concentration gradient of hydrogen ions, so you can use that to produce energy. You do have to have certain membrane proteins that are involved in something called facilitated diffusion, because these are things that cannot cross the membrane on their own. So that would be like an aquaporin, which will allow water across. You can have some passive glucose transport, and you can have some channels that allow sodium or potassium or other ions to pass down their concentration gradient. They're just doorways to allow certain things in. So I tend to think of these transport proteins as like doggy doors or pet doors. They're gonna open up your house or whatever to specific things, but unless it's a huge doggy door, people should not be able to get in and out. And so it's got specificity. So, let's see. this is not the easiest thing to read. All it is, it's a summary of diffusion, facilitated transport, which is a subtype of diffusion, and then these two, active transport and bulk transport, we'll talk about in a moment. They both require energy. The top two don't need energy. The bottom two do need energy. Osmosis is another special case of diffusion. In this case, it's the diffusion of water. This water will usually have stuff dissolved in it. We're not focusing so much on the solute, which is what is dissolved in it, as we're focusing on the solvent, the thing that it's dissolved in. So you usually have a selectively permeable barrier, and the thing that can cross, it tends to be water. Anything else, usually they'll have like sucrose or another large molecule that cannot cross through a membrane or a barrier. So you're going to have solute concentration on one side will generally be higher than the other. But if you think about it, if you've got more solute on one side, you're going to have less water per molecule of solute. On the other side, you have less solute, so you have more water filling up that volume. And it's all relative. Where do you have more water? Where do you have less water? Where do you have more solute? Where do you have less solute? In general, things always want to move so that there is an even concentration at the end. Systems are always moving towards equilibrium where you have equal concentrations. So if you think about it, you have a barrier between two compartments. One of them has very concentrated liquid. I think of this in terms of Kool-Aid because it's a good mental image for myself. One of them has really dark concentrated Kool-Aid that's very strong. The other one has very light, weak Kool-Aid that is very weak. You can't taste much. If you put them together and allowed water molecules to cross, they would eventually move so that the Kool-Aids had equal concentrations, the same color, and the same strength of taste. So things are always evening out. And there is something called osmotic pressure. That is pressure that develops because water is trying to move in one direction or another. That is really important for plants, which is when we get into all that lovely talk of water potential in a number of slides. So across a membrane, when you have osmosis, you have three different types of solutions you can have. They are all comparison solutions. So I can say, hey, solution A is isotonic to solution B. I can't just walk up and say, hey, that solution's isotonic. Isotonic to what? You're saying it has an equal concentration. You're not saying what it's equal to. So isotonic solutions, if you put a cell in a solution that it's isotonic to, it will not lose or gain water. Some water may move in, some water may move out, but overall, you're not going to have any net movement, meaning if you, say, took a mass of this cell before and after you placed it in this isotonic solution, it wouldn't gain any water mass and it wouldn't lose any water mass. On the other hand, if you have a hypotonic solution, it's got lower concentration of solutes. It's more watery. So water is going to move away from it and move into cells. And we'll look at some diagrams too that should be fairly similar to ones you've seen lots of times. It causes what's called turgor pressure in plants. That's pressure pressing out against their cell walls. That's when they're happy. That's when they're actually upright and not wilted. However, animal cells cannot tolerate this condition. They'll lyse or rupture because we have no cell wall keeping our cell membrane intact. Your cell membrane can only stretch so much. Once you pour more water in, it's just going to pop. Hypertonic solutions have a higher concentration. They are hyper, like hyperactive, tonic, so they have more solute. What will tend to happen is water will move towards the higher concentration of solute, so water will leave the cell. At that point, you will have shrinking animal cells. They'll just shrink like a raisin, like a grape left out in the sun turning into a raisin. We say they are crenated. That's called crenation, not cremation. And then in plant cells, what will happen is plasmolysis, where they start to, their cell walls stay the same shape, but their cell membranes start to pull away from it because they're losing so much water. So This is kind of a more complicated version of the diagram you usually see. If you have cells in isotonic solutions, notice that animal cell is happy. The plant cell, it's okay, but it's happiest at this level in a hypotonic solution because water is coming into the cell so much that it helps put pressure against the cell wall. Remember, plants don't have bones, so the thing that's keeping them upright is turgor pressure and their cell walls. Animal cells, on the other hand, burst. That's why it would be a very bad idea to give somebody an IV filled with plain distilled water. If you do that, you're going to introduce a hypotonic solution to their blood, and all of their blood vessels will start their blood cells will start bursting. So don't ever do that if you go into medicine. On the other hand, let's say you put a really strong concentrated salt solution in their IV for reasons I don't understand. What will happen is their red blood cells will still die. They will now shrivel up, and water will start leaving them. They won't be able to maintain volume, perform a lot of metabolic processes. It's bad news. For a plant cell, notice how the plasma membrane, the cell membrane, has pulled away from the cell wall. That's plasmolysis. All right. You can also have facilitated transport. It's passive. You don't use energy, but you do have a carrier protein. So they can't pass through the cell membrane on their own. They need a doggy door, basically. And if you have a doggy or a cat door, a pet door, unless you change the settings, that pet can come in and out as much as it wants. Same thing with these molecules. They tend to not have controls. These carrier proteins will just let stuff in or out. And the direction this stuff moves will usually depend on the concentrations on inside or outside the cell. So here's an example of some facilitated transport with a carrier protein taking a solute across the membrane. And if you're wondering, well, how does that work without energy if it's changing shape? Usually the binding of that solute will actually cause the protein to change shape so that it's opened up onto the other side of the cell. It's a little crazy that proteins can do that. I'd love to find a model that can actually do that so y'all could see it, but nobody makes one. Active transport requires energy. This is what you have to use ATP for. Most of the time you are concentrating something or you're building a concentration gradient. So you are moving things actively from a low concentration to a high concentration. It's like a greedy person sitting there going, give me everything, give me everything. They're gathering everything up. And usually there are proteins that are involved in moving this stuff. And this is the biggest example, the sodium-potassium pump, which is important for the nervous system. What it does is for a round of transport, when it's open to the inside of the cell, its shape is such that it will bind some sodium ions. An ATP binds, gets used up, that's your energy that you're using, and that will cause a change in shape for the molecule